first lecture for the nervous system, talking kind of about an overview of the anatomy and physiology of the nervous system and focusing on the action potential and clinical considerations. Before we go on to the actual material, I did want to say that we're going to have a quiz in the next few days. Because I'm posting this lecture pretty late, the nervous system will not be on the quiz. It will just be on the endocrine system as well as the lymph and immune system. So I just wanted to kind of calm your nerves about that. There's kind of a lot of learning objectives listed here. I'm just going to briefly review them. This is the same thing that's posted in those bullet points above this lecture. So you should be able to recognize parts of the neuron and their function in the context of electrochemical signaling. Again, kind of getting more into function than anatomy, thinking about neurons as afferent, association or communicative, and efferent based on a description of their function. So again, thinking about physiology, not their structure, because when we're talking about the nervous system, there's a lot of ways to differentiate it anatomically versus physiologically. So we want to start thinking about the actual functional role of the nervous system. We want to kind of know the prerequisites of a sensation or a perception, understand the relationship between a stimulus, different types of membrane channels, a greeted potential, and an action potential. So basically, how does the structure of the membrane and the flow of material across a membrane in response to a stimulus cause an electrochemical signal to be sent? In doing that, we're going to be thinking about the process of depolarization, repolarization, and hyperpolarization that occurs during an action potential, and thinking about how that relates to the flow of ions across their channels. We'll interpret a plot of an action potential and identify key measurements and periods within that, distinguish between saltatory and continuous conduction, identify EPSPs and IPSPs. The E stands for excitatory, the I for inhibitory, as being associated with depolarization or hyperpolarization. So thinking about how does that electrochemical signal pass from one neuron to the next. Explain the mechanism by which neurotransmitters are released and bind to a postsynaptic neuron, so the next neuron on the other side of the synapse, and recognize the clinical significance of dopamine for Parkinson's disease, the role of the lumbar puncture for meningitis, and the cranial nerves in the context of the neurological exam. Okay, so we're starting with chapter 12, and the slides for chapter 13 are going to be very, very brief. So most of this lecture will be spent on chapter 12, the nervous system and nervous tissue. And we'll cover sections 1 through 5, starting with a basic review of the structure and function of the nervous system, get into nervous tissue, but again focusing on its function, and then getting into the action potential and how that leads to communication between neurons. So remember last week we talked about the endocrine system and how it is involved with chemical signaling. It's more of a slow response. You have these feedback loops where you're maintaining the settings within your internal environment. And we contrasted that with the nervous system, which has both electrical and chemical signaling. It's tricky because a lot of the times we say electrochemical. There's going to be some situations where the flow of ions leads to this electrical signaling. There will be other situations where we distinguish between electrical and chemical synapses and connections. So kind of a lot of both. But this is more of a fast response to something coming in from the outside, from the external environment. So we'll think about how we receive information from our environment as well, which does follow a lot of similar mechanisms to the endocrine system, but again is much faster with a different set of pathways. And it's much more specific than the endocrine system as well. Okay, so I'm going to briefly review kind of some anatomy, but also kind of prime you for how physiology is different in the context of the nervous system. So I'm combining sections 1 and 2 here. So you may have seen a figure like this before, kind of the different divisions of the nervous system. And the first way that we separate it out is by central nervous system, or CNS, versus peripheral nervous system, or PNS. And I'll talk about this on the next slide as well, but the CNS is your brain and your spinal cord, and the peripheral nervous system is basically everything else. And we kind of start to divide that according to function. So this plot has always been kind of weird to me because it's both anatomy and physiology, and that's tricky with the nervous system. But in the peripheral nervous system, we have both motor neurons and sensory neurons. Motor neurons send signals from the central nervous system, so from your brain and spinal cord, to effectors, things like your skeletal muscle, your smooth muscle, your glands that make stuff happen. That's often in response to sensory input that is sent from sensory organs to your central nervous system. Those might be coming from general senses like touch, which have receptors distributed throughout your body, or from organs affiliated with special senses like your eyes or your nose, which are much more specialized to a particular sense. So sensory is signal going in, motor neuron is signal going back out, and that might be sensory somatic, in which you're responding to something voluntarily, or autonomic, in which it's an involuntary response, which might be sympathetic, so fight or flight, or parasympathetic, so rest and digest. And we'll focus a lot more on that, on reflexes and the autonomic nervous system in the next lecture. So I mentioned that I would show you kind of the anatomy of the CNS and the PNS. So here we see our brain and our spinal cord as the central nervous system. Those are protected by our bones. So the brain is contained within your cranial cavity, the spinal cord within your vertebral cavity. But just like how the axial and appendicular skeleton have a little bit of overlap because the appendicular skeleton involves the stuff that connects those two together, the peripheral nervous system does kind of extend into your cranial cavity and your vertebral cavity to make those connections. And the nervous system is really all about those connections between neurons and between glial or support cells that send signals and allow you to have such specialized codified responses. So again, the peripheral nervous system is everything other than the brain and the spinal cord, and components of it do extend into the cranial cavity and the vertebral cavity to make those connections. So when we're thinking about the structure of a neuron, which is kind of that classical type of cell you imagine for the nervous system, we have this big cell body or soma, which does have a nucleus as well as other organelles. So this is kind of the most like our general cell types, but these neurons have a lot of specialization to them. So they have a lot of what are called processes. This is kind of tricky because a process in the nervous system and also in bones refers to a particular structure. So it's the extension of the cell, so an axon or a dendrite, something sticking out. A process, though, also in physiology refers to some series of events. So we have the anatomical sense of process, the extension of the cell, and the physiological context, which is something happening. So here with the neuron, a process means axon or dendrite. Axons refer to those fibers, those kind of long extensions that connect neurons to their targets, so postsynaptic cells. The signals that we'll talk about today, like action potentials, are sent along those axons from the cell body. Dendrites are receiving input from other neurons. So if the cell that we're looking at is postsynaptic, it's receiving information from a previous neuron on the other side of the synapse. That information is being received through its dendrites. And it's very important that there's many dendrites because a cell at any given time can receive many different signals from many other neurons that it has connections with because of all of those dendrites. When we look at myelin, that's that fatty substance that wraps around the axon and helps propagate signals in saltatory conduction. It's made a little bit differently depending on whether we're talking about the central nervous system or the peripheral nervous system. Here we can see something called an oligodendrocyte, which tells us that this is from the central nervous system. So again, just a review of anatomy, don't stress about it too much. When we're talking about clusters of the cell body, we're talking about gray matter, and when we're talking about clusters of the axons, we're talking about white matter. And if you think back to anatomy, you can actually see those clusters of gray matter and white matter when you dissect a brain or on cross sections of the spinal cord. But that white matter I want to focus on for just a minute because it speaks to this challenge of distinguishing between anatomical and physiological distinctions in the nervous system. So if we just kind of take our eyes as a case study, we are receiving information through our eyes. So vision is a special sense. It's kind of clustered there in your eyes. So if a signal is being sent through sensory neurons from our eyes to our brain, when it's on one side of this imaginary line, when it's part of the peripheral nervous system, we call it a nerve. And when it's part of the central nervous system going into your brain, we call it a tract. So this bundle of axons in the central nervous system is a tract. In the peripheral nervous system, it's a nerve. So it's the same exact structure. We just call it the optic nerve when it's in the PNS and the optic tract when it's in the CNS. So again, it's very challenging to fit structural and functional divisions together. We get a very messy kind of flowchart and plot as a result. So there's going to be a lot of different ways that we think about the nervous system and distinctions within it. So when we're thinking about functional divisions, rather than just splitting it up into CNS and PNS, we start thinking about the types of voluntary and involuntary signals that were being sent out. So the nervous system receives information about us, so sensory functions, and then in response to that, it generates some sort of response. It might just end at kind of processing that signal into something understandable and meaningful, or it might end in a motor function, maybe voluntary or involuntary. And so when we have all that information coming through, we integrate it with memories, with our emotional state, with kind of higher level cognition, again, into a more meaningful pattern that we can then choose to react to, or maybe we don't have any choice in the matter. So the sensory somatic nervous system, the SNS, involves conscious perception, kind of interpreting a stimulus, making it more meaningful, and then voluntary motor response. So here, the effector, EFF, ECTOR, the thing that kind of acts on this due to a signal from motor neurons, is skeletal muscle. It's voluntary. Whereas with the autonomic nervous system, the effectors are smooth or cardiac muscle and glandular tissue. So this is involuntary control of the body to maintain homeostasis, the autonomic nervous system, or ANS. So again, I've kind of spoken to this point that for a sensation to occur, you have to have some prerequisites. So you have to have a stimulus first, and we'll talk about this a little bit more in a bit, but that stimulus has to be present. You can't just have this signal being sent out of nothing. You have to have reception, so you have to have a receptor picking up that stimulus. The receptor has to match the stimulus, and then it's able to convert it into a nerve impulse. We'll explain the mechanism for that in just a moment. That nerve impulse is then conducted to the brain, so it has to keep going along a series of neurons to get to the central nervous system, and it has to be integrated and perceived into something kind of more meaningful. So you translate that pattern, you have perception, you have a meaningful pattern or sensation. So integration leads to perception and sensation from a stimulus. So oftentimes we end the functional and anatomical divisions of the nervous system at the ANS and the SNS, but there's also something called the enteric nervous system, which is kind of overlapping with the autonomic function of the nervous system, but it's also kind of its own thing. Your gut has a lot of nervous function, so it's considered your second brain. It's actually capable of integration. So here's just kind of a picture of how complex this system actually is. And if we kind of rethink those divisions of the peripheral nervous system, we kind of get something a little bit more balanced. So we have motor and sensory being equal. Before, sensory was kind of off on its own and motor took up the bulk of the chart, but it all really is feeding in together. So that motor section has signals being sent along, skeletal and kind of more intentional responses, and then also autonomic. That might be sympathetic, parasympathetic. There's the role of the gut, and we have kind of signals being sent in a lot of different directions. Okay, so to get back into the function and the physiology of the nervous tissue, this is the slide that we already kind of unpacked, so I'm just going to zoom in on different parts of this slide and talk about the function of other components and kind of set us up for talking about the action potential. So remember that the goal of the neuron is computation and communication within the nervous system. So it needs to be able to receive and understand and process information and communicate that information to other cells and to the central nervous system. So, and then back out through the peripheral nervous system. So it's electrically active. It's able to release and propagate electrochemical signals to target cells across synapses. And what we mean by the synapse is a specialized area of contact between two neurons. So going from the kind of end point of one neuron to the cell body of the next neuron. And so the dendrites are going to receive, the dendrites on the next neuron, on the postsynaptic neuron, are going to receive information from those neurons at that point. And so if we kind of zoom in on the middle portion and think about that signal being sent and how to make it more efficient, here we see a couple of different structures. We have first, the signal is being generated in the cell body. Again, we'll talk about what actually causes that signal to be sent, how it initiates, but it's spreading from the cell body to the axon. And so it travels first to this initial segment or axon hillock where the cell body tapers and you go towards the axon. I want to point this out because this transition from cytoplasm to axoplasm is very important because the contents of that axoplasm are what really set us up for the action potential for that electrochemical signal, because there's a very particular balance of ions in that axoplasm. So moving away from this cell, which has its own function, to this unique axoplasm, which has a very different function and a very different relationship to the surrounding environment, is going to be important. We also have these nodes of Ranvier, which are important for saltatory conduction, for jumping the signal ahead. That's related to those myelin sheaths. Sorry, I skipped ahead. Let me see if I have this here. Okay, sorry. So the myelin sheaths, again, are kind of that fatty material, and that combination of the fatty insulating material and then those open nodes of Ranvier allow that signal to be sent ahead due to the insulation. So again, it goes from that initial segment to the axon terminal, which is the end of the axon, and there are several branches there extending towards target cells, ending in synapses and synaptic clefts. So when we see the synaptic clefts, there's a synaptic end bulb, which is this enlargement. You'll see that when we talk about synapses in a little bit more detail, just in terms of the structure. Okay, so remember back in anatomy, we talked about how neurons can be classified by shape. So unipolar, bipolar, multipolar, don't stress about that for physiology. What you should think about instead is the physiological classification of neurons. So when we talk about afferent or sensory neurons, these are sending signals or nerve impulses from receptors to the central nervous system. So they're sensing, they're perceiving information about the world around us. Well, not quite perceiving it yet. They're sending information from the world around us, sensing, to our central nervous system. When we have connections from neuron to neuron, that's called association or connecting neurons. And then the efferent neurons, our motor neurons, have an effect. So efferent effect, these send information from the central nervous system to our effectors. So the skeletal muscle or smooth muscle or glands. Skeletal muscle in that somatic sensory nervous system, smooth muscle and glandular tissue in that autonomic nervous system. So to kind of picture that a little bit more clearly, we have an afferent nerve sending signals from the skin where we have touch receptors to our central nervous system, the spinal cord. Information is sent back out from the spinal cord along efferent nerves to muscle in response. So in this case, maybe your skin is pressed up against something very hot, and so you're receiving information about that and then sending that information back out through efferent nerves to your muscles so that you can move your arm away from that hot object. There's a lot of other types of cells in our nervous system other than neurons. So there's these glial cells, also called neuroglia. There's support cells like astrocytes, microglia, the different cells that produce myelin sheaths. But we're not going to go in depth on them. I am posting anatomy slides separately. I think they're already posted. They should be. So if you want to review those glial cells, you're welcome to if you think it'll help you, but we're not going to go in depth on them for this lecture. So the importance of talking about the anatomy of neurons was really to set us up for the physiology of neurons and the action potential, understanding how these signals are sent from different parts of your body to other parts of your body. So when we're thinking about sensation and sensory receptors, sensation is really just the activation of sensory receptors by a stimulus. It's that first step. And it doesn't really become something that we can recognize until we have perception. So that central processing of the sensory stimulus into a meaningful pattern. After we have perception and integration, then we might have a motor response when we're talking about the somatic system, when we're talking about a voluntary response. So these signals are being sent from sensory cells to the central nervous system. We have integration and then a motor response if we need to. So when we're thinking about that stimulus and receptor, it's really a similar process for a lot of senses as when we're talking about the endocrine system. So we have some structure binding to a receptor on a target cell. We have a series of transduction, maybe we have different channels being opened, and we have some cellular response if we meet a certain threshold in a lot of cases. So like I mentioned, I've shown you this slide before, but I want to keep reiterating this point that we are not constantly consciously perceiving information about all of the information about the world around us. Remember, we have to have that reception of a stimulus. We have to have a physical connection being made between the stimulus and the cells of our body, and that physical connection then has to be converted into something that can be sent to our brain for processing and for integration and perception. So we have to have this series of requirements being met before we can actually perceive that as something conscious and then react to it. So in this classical sensory receptor motor response pathway, we have our sensory endings on our skin reaching underwater. A signal is sent along an afferent pathway into our central nervous system. It's processed and then sent back out through an efferent pathway back to our skeletal muscle so we could possibly move our hand if we wanted to. So when we're looking at these sensory receptors, we have to think about what is actually causing that afferent signal to be sent. So remember, we're not always sending afferent signals for every single stimulus. There has to be a connection between the stimulus and our cells. So if you think back to last week, remember we talked about cyclic AMP. We talked about how a hormone binds to an extracellular receptor, which has a G protein. We talked about that series of activations and about protein kinases and phosphorylation cascades to have a cellular response. So it's very similar here. If we take, for example, shifting from touch to specifically thinking about smell, here we have a stimulus, which is an odorant, like from a plant, something you can smell. It's binding to a receptor within our nose, which causes transduction. So in this graphic on the right side, you can see that cyclic AMP being activated. You can see ion channels opening. Then that changes the cellular environment. So from now on, we're going to be talking a lot about how ions affect that internal environment of the cell. And remember, I talked about how transitioning from cytoplasm to exoplasm is really important because the contents, that cellular material, and having very specific resting membrane potential, which I'll get into in just a moment, is going to be really important. I'll define depolarization in just a sec, but you should start to be thinking about this idea that the connection between a stimulus outside the cell and a receptor on the cell might result in a change to the cell environment, which is then passed on from cell to cell. So that stimuli or the stimulus might affect a membrane potential to different degrees depending on the stimulus. So if we look at this plot, we have time, and then on the x-axis, and then on the y-axis we have what's called membrane potential. So I will kind of expand on that in just a moment, but for now you should just kind of think about the size of that as being related to the size of the stimulus. So a couple things to note is that we have a couple of these kind of smaller stimuli, but then we might have one that crosses what's called a threshold. So that threshold, we should start thinking about the idea that there's a tipping point where maybe we have a much stronger reaction that can be sent to other neurons. Another thing to note is that when you have a longer duration stimulus, it's not necessarily going to increase the size of the stimulus. So we have kind of differences in intensity versus duration, so you can kind of like get used to things. If you have something resting against you, you might kind of perceive it at first, but you adapt to things and readjust. But if you have kind of repeat signals being sent, that frequency does have an effect. So we'll unpack that in a little bit. But I just want you to be aware of this idea that there's different stimuli. Some of them might have an effect, some of them might cause these changes in the internal cell environment that then get translated into an electrochemical signal. Others don't have that same effect. So again, that stimulus has to cause the membrane potential to pass what's called a threshold in order to generate an action potential. The actual number of the threshold does matter, and so we'll talk about that in just a moment. All right, so when we're thinking about action potentials, this process by which an electrochemical signal is being sent through our neurons, it really depends on stuff moving through the cell membrane. And remember, when we have a cell membrane, we use the fluid mosaic model to describe it and study it. We have this phospholipid bilayer where there's the hydrophobic tails in the center. So that kind of limits what's able to pass through the cell membrane. We do, however, have a bunch of proteins embedded in that membrane, including channel proteins, which have pores. This one is kind of pictured as just being open. They're not all like that. But what's really great about channel proteins is hydrophilic molecules like ions can really easily pass through those channel proteins through what's called facilitated diffusion. So it's a passive process, but you do need the channel protein present. There is some selectivity with channel proteins, so these can be charge specific, which is called electrochemical exclusion. But it's not just as simple as having an open channel protein, so I'm going to talk about a few different types of membranes which play a role in these signals that are sent. The first of which are sodium-potassium pumps. So this is where a lot of the ATP that's generated in your body goes to power these sodium-potassium pumps. I'm going to let this GIF play for a little bit, and I'm going to kind of unpack it verbally and then kind of put up a series of steps that explain it. So here we have that cell membrane. So we have the little blue tails, which are the hydrophobic tails. On the top part of the membrane, so above the membrane, that's the outside of the cell, and those little circles are meant to represent the sodium ions. On the bottom of the membrane, we have the inside of the cell, and those triangles represent potassium ions, so K+. And so we see that this pump, which is not a channel protein, it's a pump, it requires ATP, it's active transport. We see that it's moving, we see that it's binding to these different ions. And so what's happening here, we have it, let me let it restart. So we have the sodium binding, 3 sodium, and in the bottom right, you see that there's a P there, and that P represents an ATP, so adenosine triphosphate being cleaved and forming a DP, and using that cell energy to power this pump. So the sodium is being pumped out of the cell against its own concentration gradient, which is why it requires energy. And then the potassium is being pumped into the cell. So again, against its own concentration gradient, requiring energy. So step-by-step process here, the sodium concentration is higher outside the cell, potassium is higher inside the cell. Both of these ions get pumped against their concentration gradient to build it up further. 3 sodium ions bind to the sodium-potassium pump, 1 ATP binds, that ATP is split to move sodium outside of the cell against its concentration gradient. The enzyme changes shape, and then potassium binds, that's located outside the cell, and it's pumped inside. So the reason we spend so much energy to keep these guys going is because that charge difference that's established represents potential energy. It keeps these concentration gradients really strong so that we can use them to our advantage when we want to send signals. So these sodium-potassium pumps, along with these guys, leakage channels, are really important for contributing to resting potential, for keeping that charge difference the number it's supposed to be at. So leakage channels are not intentionally controlled. They're just like randomly open or close at a given rate. And so that random opening and closing and the intentional action of the sodium-potassium pumps maintains that charge difference, that resting potential. There's also ligand-gated channels, which open when a neurotransmitter binds to them. They're extracellular, so we have something like acetylcholine maybe, that's that ACH binding, and then those are going to cause ions to move along their concentration gradients. So we call these receptors ionotropic because that binding of the neurotransmitter leads to a movement of ions. There's also mechanically gated channels, which open when you have a physical change on the plasma membrane. So for example, with somatosensation or touch perception, we have the plasma membrane being distorted. That causes that mechanical gate to pop open. Ions move along their concentration gradients, and that's going to start this depolarization. So it's going to start that change in charge, which gets the signal going. Finally, there's voltage-gated channels, which are very, very crucial for the process of action potentials. So these respond to changes in the electrical properties of the membrane. And if we look in this bottom left, we see in this image when the voltage-gated channel is closed, the voltage inside the cell is measured at -70 millivolts. So that number is very important because that is the resting membrane potential. That -70, you need to know that. And here we see that when this voltage-gated channel opens, it's at a voltage of -50. That is also important because it turns out that the threshold it needs to meet in order to open up is -55. So we're going from -70 past -55 to -50, and we have these voltage-gated channels opening. Now ions are able to move along their concentration gradient. So they've been kept separate, they've developed this strong concentration gradient. Now they're past this particular voltage, these channels open up and they move along their concentration gradient and cause a dramatic chemical shift. So another thing to keep in mind is that those voltage-gated channels don't open up and allow all ions to move along their concentration gradients. There's ones that are selective for sodium ions, others that are selective for potassium ions. So I wanted to define some of these terms that I've already kind of been using and introducing. When we're talking about depolarization, this means becoming more positive and less negative. So remember, resting membrane potential is -70. So when we move away from that to become more positive or less negative, that process is depolarization. And when we depolarize to a certain point, we pass that threshold, we start opening those voltage-gated sodium channels. Those sodium ions are able to rush inside the cell, and the inside of the cell becomes more positive. So that's kind of a positive feedback loop in a sense. We have, that movement towards depolarization that opens up those sodium channels, and it's an even faster depolarization process. But there is a peak that is reached, a certain voltage at which those voltage-gated channels close, and you have repolarization, which is returning to being more negative, returning to having these strong concentration gradients across the cell membrane. So the sodium channels close, the potassium channels open, and those potassium ions are then able to rush outside of the cell, and the inside of the cell becomes more negative. And it actually kind of overshoots a little bit. So you go through this process of hyperpolarization where this inside of the cell becomes even more negative than the resting potential. But then as you have those sodium-potassium pumps working and those leakage channels working, they restore those resting concentration gradients. So to kind of visualize this, the top image is showing the membrane at its resting potential of -70. This yellow channel is the voltage-gated potassium channel. The blue channel in the center is a sodium-potassium pump, which requires ATP. And the purple channel on the right side is a voltage-gated sodium channel. In this top image, you can see all of the purple sodium ions with a high concentration outside the cell. Inside the cell, you see a very high concentration of those yellow potassium ions. So one thing that's kind of tricky is that here we're talking about two sets of positive ions, the sodium and the potassium. So it's like, why is it negative at all if there's just a ton of potassium ions? But remember that a lot of cellular components, so like DNA and different proteins inside of your cell are negatively charged. And there's also just a lot of other ions kind of floating around. So it turns out that the sum of this inside of our cells is -70. So at that resting potential, the sodium-potassium pumps are maintaining the resting potential and the voltage-gated channels are closed. But when we have that initial kind of ion cascade or different signals being sent in, we have some depolarization past a threshold. That depolarization opens those voltage-gated sodium channels. So on that far right side of the middle image, we see those channels open now. The sodium-potassium pumps are no longer working, and because there's no point in spending energy to kind of fight this process. So we have sodium rushing into the cell, and then that's going to peak at a certain point. The channel is going to close, and instead and kind of around that time, the potassium channels are going to open. So repolarization occurs, voltage-gated sodium channels are closed, voltage-gated potassium channels are open. We have hyperpolarization happening, and then the sodium-potassium pump starts working again to restore the balance. So if we kind of think about all those pieces and visualize positive and negative charges on an actual axon, on this left image, we have the cell body, the soma, and we have an extended axon. And we're just going to think about kind of continuous conduction. We're not going to worry about the myelin sheaths right now. So we have the direction of the action potential going from the cell body to the axon terminal. In part A up here, we have this first part of the axon depolarizing. So let's imagine we have kind of a receptor, a signal being sent in. We have that cyclic AMP cascade happening, and we have all those ion channels opening, and we have depolarization at the cell body. So then that change in the cytoplasm and the axoplasm reaches that initial axon segment, and depolarization is happening. So it's positive, and then that passing of the threshold, that rapid release of sodium ions into the internal environment of the cell is going to cause the next set of of channels to open. So the next set of voltage-gated sodium channels opens, and that next set is then depolarized. While that's happening though, there's repolarization of that initial segment. So it continues along that way with depolarization causing the next set of channels to open and undergo depolarization of that area, and then that depolarized section becomes repolarized and then eventually goes back to resting. So we see that much more smoothly in the GIF on the right. We see that transition from positive to causing the next set to be positive and then going back to negative resting potential. Okay, so when we're thinking about an action potential, we can define that as an electrical signal that nervous tissue generates for communication. And if we kind of integrate that image that we saw on the previous slide with the other GIF that we saw on the previous slide, we get something like this. We see that when the signal is first sent from the dendrites through the cell body to that initial segment, it's going to go from negative to positive as we have depolarization, and then repolarization, hyperpolarization, and back to rest. And if we start to measure that and think about those numbers that we thought of before, like -70 and -55, as well as +30, we start to get a plot that looks like this. So we see this rapid depolarization up to a cap of 30 millivolts, and then back down to hyperpolarized at about -90 millivolts, and then back up to -70. And that kind of depolarization, hyperpolarization, and repolarization happens in a very distinct pattern along that axon segment. So we see that action potential kind of moving along. And if we kind of start to measure the different ions that are involved in this process, we see that it's a little bit messier than this. So this plot is showing the kind of movement of different charges of different ions. So we see it threshold and peak and rapid depolarization with the signal being sent out. I'm going to let the GIF restart one more time so you can kind of visualize it. So when it starts over, we have the slow increase and then past the threshold, rapid depolarization, signal sent out, and then it's going back to rest. And so all of those messy different sets of ions have to get back to where they were before in order for another signal to be sent out along this neuron. So again, we get this plot that we are used to seeing. -70 is the resting membrane potential, -55 is the threshold. After passing that, you have rapid opening of those sodium channels, depolarization, repolarization, hyperpolarization, and then back to resting potential. So -70 millivolts is the resting potential, that's important to know. -55 is the threshold potential, and 30 is the peak of the action potential. It really doesn't get higher than that. So here we see that there's a kind of different points where those voltage-gated calcium, or sorry, no, I keep saying calcium because it's K. My brain is not dealing well with chemistry right now. Potassium. So those voltage-gated potassium channels open a little bit after the sodium channels, and then they remain open a little bit later. And so we'll get back to that in just a moment. But first, it's important to remember that action potentials are all or nothing. They are always peaking around 30 millivolts. They don't increase in magnitude in proportion to the strength of the signal. So when you have those graded potentials, those do vary, but once you get past -55, you shoot up to 30 and then it peaks. So it's not going to get higher than that to reference it being stronger. But if there is a stronger signal, you do get more frequent action potentials. So it's the difference between like a loud signal like beep versus beep, 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 beep. So it would actually be the beep, 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 beep to represent something very strong as a signal. So increase in frequency, not magnitude, in proportion to the strength of the signal. Okay, so I mentioned that those ions kind of move at different rates. So we have the sodium and we have the potassium opening at different times and moving at different times. That's really important for going through depolarization and then repolarization and hyperpolarization. And it's due to differences in their structure of those voltage-gated channels. But we have to wait until all those ions are back to their normal concentration at resting membrane potential before another signal can be sent. So this is what's called the refractory period. We have the absolute refractory period where the bulk of the action is during the action potential. But then as we go through hyperpolarization back to resting membrane potential, we have the relative refractory period until everything balances out again. So I've also mentioned the difference between continuous conduction and saltatory conduction. When we have no myelin sheaths, when we have just this empty axon, that signal has to make its way through every single segment of that structure. But when we have insulation in the form of myelin sheaths, that depolarization and repolarization skips ahead between the different myelin sheaths on those nodes of Ranvier. It doesn't have to happen in a continuous sequence. So far we've been focusing on stimuli that result in depolarization, in becoming less negative and more positive. But some stimuli are inhibitory. They might lead to even more hyperpolarization. And so these signals that are being sent in happen at dendrites. And so we have dendrites receiving information. We have a stimulus being sent through the cell body and then to the initial segment of the axon. We have what's called a presynaptic cell and a postsynaptic cell. The presynaptic cell is the one that's receiving the stimulus, and it's going to possibly send a signal to the postsynaptic cell. It might pass on the action potential, but that information that's received at the postsynaptic cell doesn't always translate into an action potential. So whether it gets propagated kind of varies depending on some different information. So when we have depolarizing graded potentials, that's associated with ion channels opening to kind of allow a flow of positive ions into the cell. So if we have, for example, those sodium channels, or this time actually calcium channels, and we have a strong concentration outside the cell, if those channels open, it becomes more positive inside the cell and becomes more depolarized, so that's more stimulatory or excitatory. Hyperpolarizing is associated with positive ions leaving the cell. Remember that the concentration of potassium inside the cell is stronger than outside. So if those channels open, potassium leaves the cell, it becomes even more negative, more hyperpolarized inside the cell. Or sometimes chloride, which is a negative ion, can enter the cell and make it more negative. So we might have stimuli that produce depolarization, depolarized graded potentials, or hyperpolarized graded potentials. And again, whether that information is passed on to the next cell kind of varies on a lot of different things. So the postsynaptic potential, PSP, represents the graded potential in the dendrites of a neuron that is receiving information, receiving the synapses from other cells. So a signal is being sent from one neuron to the next, and we now have to look at the postsynaptic potential to see kind of if we're going to keep this signal going. So when we have an EPSP, that's excitatory postsynaptic potential, that is associated with depolarization moving towards the threshold, so getting closer to having an action potential. IPSP, inhibitory postsynaptic potential, is moving away from the threshold, so hyperpolarization. excitatory PSP associated with depolarization, inhibitory PSP associated with hyperpolarization. So remember, when we're thinking about a presynaptic and postsynaptic neuron, it's not just a one-to-one relationship. And neuron has a lot of signals coming in through all of those dendrites. So it might be receiving information from many different neurons that can be combined in different ways to reach threshold. And this process is called summation. So the joining of different excitatory PSPs and inhibitory PSPs may move us towards or away from threshold. So in this example, we have this kind of segment A where we have a lot of EPSPs and IPSPs, but eventually we have this one EPSP that pushes us above threshold, and then we have this rapid depolarization and action potential. So here we have kind of this combination coming together through summation in order to get that signal, that threshold being reached and a signal being sent. So that summation can be spatial, it can be a lot of different neurons sending different signals and kind of summing them together at a postsynaptic neuron, or it can be temporal, so we can have one neuron sending very rapid signals to its postsynaptic neuron, and those different signals can be summed together through summation to reach the threshold. So again, when we have communication between neurons, it's happening at the synapse. The presynaptic cell is sending the signal and the postsynaptic cell is receiving the signal at the synapse. And when we talk about synapses, they can be a little bit different in structure. So here we have a gap between them. Sometimes that gap is filled in the case of chemical synapses. In the case of electrical synapses. So with electrical synapses, we have a gap junction that connects them and actually allows ions to flow through. So it just continues that depolarization, that rapid change. So here in that presynaptic cell, we have those presumably sodium ions flowing into the cell, changing the memory potential, causing it to become more positive, and that's just going to continue along through that gap junction into the postsynaptic cell and keep that process going. With a chemical synapse, we have the release of chemical signals, which are called neurotransmitters. So here we have the synaptic cleft, that kind of widening. We have the synaptic cleft is the open space. We have the synaptic bulb, which is the widening, and we have these vesicles that are holding neurotransmitters that are about to be released across that synaptic cleft. And we see that there's receptors for those neurotransmitters at the postsynaptic cell. So I'll talk about that mechanism next. So those voltage-gated calcium channels are really important for this neurotransmitter release. When we have the action potential arriving at the synaptic bulb, that causes those voltage-gated calcium channels to open. So that's going to change the charge inside of this synaptic bulb. It's going to cause all these vesicles that are full of neurotransmitters to move towards the edge of the cell, go through exocytosis and release those neurotransmitters into the synaptic cleft. They then cross that synaptic cleft and bind to those receptors. So they travel across, they bind to very specific receptors. There might be a series of receptors there, but you have to have the correct receptors for your particular neurotransmitter, and they can't be blocked already by different medicines or drugs or existing neurotransmitters. Another important thing that you have to do is now you have all these neurotransmitters kind of floating around. They have to be broken down or moved out or recycled in some way. So they either diffuse out, or you have enzymes coming in and breaking them down or moving them around, or you have reuptake of them back into the neurons. So when we're thinking about different neurotransmitters, there's a lot of different groups of them. One of the most well-studied is acetylcholine, which is part of what's called the cholinergic system. There's amino acid or small molecule neurotransmitters, Other types of small molecule neurotransmitters include purinergic, so things like adenosine, as well as biogenic, things like dopamine, and then the neuropeptides. So these are kind of series of amino acids held together to form a polypeptide chain. I mentioned that acetylcholine is one of the most well-studied of these, and it was used in an image we saw on an earlier slide to kind of cause changes in ion concentrations. So acetylcholine is released in the PNS as well as some central nervous system neurons. It's really important for the neuromuscular junction, which you should have talked about in anatomy. It can act either excitatory or inhibitory depending on the neuron and the receptor. And something that's kind of interesting about it is that it binds to both nicotinic and muscarinic receptors. Those bind also to nicotine and muscarine respectively, which are two types of drugs, but they can't bind to each other. So muscarine can't bind to the nicotinic receptors, nicotine can't bind to the muscarinic receptors, so there's kind of unique binding properties there with acetylcholine. There's also the catecholamine neurotransmitters. So this is another term for norepinephrine, epinephrine, and dopamine. These are modified versions of the amino acid tyrosine, which you see pictured up above. Then there's the chemical structures of dopamine, norepinephrine, and epinephrine, and you can see that they are very similar. They're just biogenic, they're biogenically modified versions of that tyrosine. And again, they can be excitatory at some synapses and inhibitory at others. Okay, so I know that was a lot of information. For chapter 13, I'm just going to briefly talk about a few case studies. Thinking first about a case study of the central nervous system, some clinical applications of different parts of circulation in the central nervous system, and then briefly introduce cranial nerves with the peripheral nervous system. So you might have heard of Parkinson's disease, which is a neurodegenerative disease, meaning that it affects kind of already developed nervous systems. So it's more common in older individuals. So it causes the death of neurons, specifically those that release dopamine, which is a neurotransmitter, remember, in the basal nuclei. And so when you don't have that supply of dopamine, that leads to problems in regulating movement commands, which causes tremors and a lot of the symptoms associated with Parkinson's disease. But if you can get kind of dopamine precursors in past the blood-brain barrier, then you can start to deal with Parkinson's disease. So if you can take medication that is able to transport something like L-DOPA, which is a dopamine precursor, past that blood-brain barrier, then you can start to use it as a treatment. But it does kind of have to be caught pretty early, and there's some serious side effects associated with taking L-DOPA and other dopamine-associated medication. So that was the first case study. Now we're going to think kind of about meningitis and the blood-brain barrier, kind of this idea of the relationship between blood and circulation versus how circulation occurs within the nervous system. So remember, we have something called the blood-brain barrier, which is kind of formed in part by astrocytes as well as endothelial cells with tight junctions. So we have capillaries and junctions that are more selective, so less leaky than they might be in other parts of the body. So material can't easily pass through the way it sometimes can in other locations, depending on the type of capillary you have. There are certain medications that can pass through. For example, lipid-soluble drugs can pass the blood-brain barrier. And when you're inflamed, when you have inflammation, that allows drugs to pass through more easily. So water-soluble drugs that wouldn't normally be able to cross the blood-brain barrier can cross more easily when your body's going through inflammation. Part of the problem, though, is that your immune system is privileged, so it doesn't necessarily react to antigens the way that other parts of your body might. So remember, we have different layers. We have of the meninges, we have the dura mater, which is attached to bone. We have the arachnoid mater, and then the subarachnoid space, which has cerebrospinal fluid, and then the pia mater, which kind of goes over your cerebral cortex and the outside of your brain. So it kind of forms those layers between the bone and your brain. And then it extends into your spinal cord, or along your spinal cord into your spinal cavity as well. Vertebral cavity, sorry. So then when we're thinking about this, there is kind of a portal of entry into the central nervous system here because we have the formation of that cerebrospinal fluid and filtration and circulation through there. So when we have inflammation, not only does it allow drugs to pass through more easily, it allows microbes to also pass through into the cerebrospinal fluid a little bit more easily than they normally would. So we see that here, that relationship between the capillaries and the cerebrospinal fluid. So when we're thinking about meningitis, that's swelling or inflammation of the meninges. So meningitis, itis means swelling or inflammation of the meninges, meningitis. So this might cause fever, headache, stiff neck, but can also progress to nausea, vomiting, convulsions, and coma, and possibly death. And actually, in a lot of bacterial cases, it can lead to death quite easily. The bacterial cases are a lot easier to treat because we have antibiotics, but with viral meningitis is a lot more common. It's harder to treat, but it's less fatal. So viral meningitis is harder to treat, less fatal. Bacterial is easier to treat, but much more fatal. And there's three main causative agents of bacterial meningitis: Streptococcus pneumoniae, Neisseria meningitis, and Haemophilus influenza type B, which you should have vaccinations against. There's also a lot of other causative agents, though, as many as about 50 bacterial species that can opportunistically cause meningitis. So, for example, we talked about HIV and AIDS and immunocompromisation a couple weeks ago. If that's the case, then a lot of other things can cause meningitis as well. And I know a lot of us have children, and we're warned when we're pregnant not to eat certain foods because they might cause listeriosis, or they might have listeria. So things like cold deli meat that's been processed, or processed salads, different things like that that are prepackaged. So that causes kind of flu-like, or not really flu-like, but very mild foodborne illness in the adult, but in fetuses and infants, it can cause meningitis and it can be fatal. So the meningitis and swelling of the meninges might not necessarily affect the adult, but will affect the fetus. So when we're diagnosing meningitis, we have to take a sample of cerebrospinal fluid. And so we're able to kind of look at different profiles and determine what type of meningitis the patient has. Because remember, it can have many causative agents, and if it looks aseptic, it might be viral, and then we can't really figure out how to treat it, but again, it's not as fatal. So the way that that's done is through a lumbar puncture. Remember, lumbar is like the lower back region, those lower vertebrae and the lower spinal nerves. And so a needle is inserted into that lumbar region of the spinal column. Cerebrospinal fluid is extracted and tested to get that clinical profile. Okay, so and then for the last case study, I wanted to briefly talk about the neurological exam and cranial nerves. So again, thinking about what causes that afferent signal to be sent and the pathways along which they are sent. So remember, nerves are bundles of axons in the peripheral nervous system. In the central nervous system, they're called tracts. So this is the white matter. And when we have cranial nerves, these are nerves that are directly attached to the brain and are responsible for sensory and motor functions, primarily of the head and neck, and they're associated with a lot of sensation. So there's 12 of them total. There's the olfactory, optic, oculomotor, trochlear, trigeminal, abducens, facial, auditory or vestibulocochlear, glossopharyngeal, vagus, spinal, accessory, and hypoglossal nerves. For some classes, these are important for you to memorize, so I just wanted to kind of reiterate them. I will not be testing you on them because you have more than enough to study for this exam, but I just want to point out that some of these are sensory, some of them are motor. So again, in the next lecture, we'll be talking a little bit more about reflexes and about the autonomic nervous system, so kind of getting more into that level of organization and kind of thinking about the signals that are sent. That's it for now. I'm sorry, I know that there's so much information for this exam, and I know that we're scaling up a lot from the last exam. I really wanted to give you a chance to kind of adapt to the course and develop your study habits, but now we do have to get through a lot of material. It is physiology, and I do need to prepare you for things like the TEAS test and expectations of you in nursing and other health professions. So I'm just thinking about the study guide and how much you guys have to do. I'm really trying to do my best to make sure you're prepared for it. So I have the study guide mostly prepared, and I will post it for you by 5 p.m. tomorrow, so Monday, the 14th. Please email me as usual if you have any questions, and hopefully things are going well.